Imagine you fall asleep tonight in your own bed and wake up 50,000 years ago. It is cold and quiet. There is no city noise, no phones, no cars, only a small group of hunters and gatherers around a fire. One of them turns toward you. Their hair is tangled, their clothes are made of animal skins, but the face, the hands, the body proportions look familiar. Give this person a haircut, a t-shirt, and jeans, and they could probably walk down a modern street without attracting much attention. This is one of the striking facts we know from anthropology. Anatomically modern humans, people whose bodies looked very much like ours, already existed tens of thousands of years ago in Africa and had begun to spread across the world around 70,000 to 50,000 years ago. According to the widely supported Out of Africa model, most people living outside Africa today descend from those ancient travelers. In other words, the basic blueprint of our bodies was already in place long before history, cities, or science appeared. If our bodies were already so similar 50,000 years ago, did humans at some point simply stop changing physically? Did evolution press a kind of pause button once Homo sapiens reached a successful design? Or are we still changing, just in ways that are less obvious than a bigger skull or a differently shaped jaw? In this story, I want to show that humans did not stop evolving. Instead, what changed was the way evolution works on us. Our environment shifted from mainly natural, climate, predators, disease, to something much more complex, culture, technology, and global societies. Tools, clothing, houses, and cooking changed how our bodies experienced cold and hunger. Agriculture altered what we eat and how we live. Modern medicine influences which of us survive and have children. All of these forces still shape the human body, but often quietly, in our genes and in statistics, rather than in dramatic new body shapes. As we move on, we will travel from the distant past to the present and a little into the future. We will look at how our species became anatomically modern, what scientists mean when they say evolution has slowed down or changed direction, and we will examine clear examples showing that physical evolution is still going on right now. Finally, we will ask what these changes might mean for people who will live 50,000 years from now, and whether they would look at us the way we look at those ancient humans by the fire, familiar and yet living in a completely different world. Before we dive in, drop a comment and tell me where in the world you're listening from. I'd love to see how far this story has traveled. People sometimes say that humans stopped changing physically 50,000 years ago, but that sentence only makes sense if we are clear about two everyday words: physical change and evolution. They sound simple, yet in normal conversation we often use them in a very loose way. In this story, I want to use them a bit more carefully. Let's start with physical change. Human bodies change all the time, but not every kind of change is evolution. It helps to imagine three different levels of change. On the first level, there is change within one lifetime. A baby grows into a child, then a teenager, then an adult. Bones lengthen, muscles grow stronger or weaker, Hair changes, skin ages. A person can gain weight, lose weight, build up their body in the gym, or lose muscle during illness. All of this is real physical change, but it happens to one individual. It is guided by the genes that person was born with, plus nutrition, lifestyle, and accidents. However, these changes do not rewrite their DNA. They do not directly change the genetic information that will be passed on to their children. On the second level, there are differences between people and between populations. When we look around us, it is obvious that humans do not all look the same. Some people are tall, others are short. Skin, hair, and eye color vary. Faces have different shapes. Body proportions, for example, leg length compared to torso length, are not identical in everyone. If we compare large groups from different parts of the world, some patterns appear. On average, people in one region might be taller or have darker skin, or have bodies that are slightly better suited to hot or cold climate. These differences are partly the result of environment and culture, what people eat, how they live, but some of them are also influenced by inherited genetic variation. On the third level, there is long-term evolutionary change in our species. Here we are not comparing one person to another, or even one modern population to another. Instead, we compare humans who lived at very different times in the past. We ask, did our ancestors, hundreds of thousands of years ago, have the same body proportions? Was the shape of their skull like ours? Were their teeth and jaws the same size? When people say that humans stopped changing physically tens of thousands of years ago, this deep, long-term level is usually what they have in mind. So in this story, when I ask whether humans stopped changing physically 50,000 years ago, I am not asking whether teenagers still grow or whether people still change their body in the gym. I am asking whether the long-term patterns in human bodies, the kind of changes we see when we compare skeletons separated by thousands of years, have really become stable, or whether evolution continued to shape them after that point. This leads us to the second word, evolution. In modern biology, evolution is usually defined as a change in the heritable characteristics of a population over successive generations. In population genetics, this can be described more technically as a change in the frequency of genetic variants, called alleles, in a population over time. The exact wording differs slightly between textbooks, but most definitions share the same basic ideas. A useful way to think about evolution is to break it into three key components: variation, inheritance, and differential reproductive success. First, variation. Individuals in any population of humans are not identical. They differ in height, in facial features, in how easily they gain weight, in how fast they can run, in how resistant they are to certain diseases, and in many other ways. Some of these differences are mainly environmental, but many have a genetic component. Second, inheritance. Some of these differences can be passed from parents to children through DNA. If a trait has a heritable basis, then over many generations, that trait can become more or less common in a population, depending on what happens to the individuals who carry it. Third, differential reproductive success. Not everyone survives to the same age, and not everyone has the same number of children. In the past, people who happened, even slightly, to be better suited to their environment often had a higher chance of surviving and leaving descendants. This might be because they tolerated the local climate well, found food more efficiently, or were more resistant to local infections. If those advantages were linked to heritable traits, then over time, those traits tended to spread in the population. This process is called natural selection, the differential survival and reproduction of individuals with different heritable traits. Natural selection is not the only evolutionary process. Random events can also change gene frequencies in small populations. This is known as genetic drift. Migration brings new genes into a population and removes others. But for our question about physical change, natural selection is especially important because it connects the environment, climate, diet, disease, lifestyle, with the way bodies are built and function. It is also important to separate evolution from development. A single person growing from a baby into an adult is not evolution. Evolution happens when, over many generations, the typical way babies grow and develop begins to shift because the genetic instructions in the population have changed. The timescale is different. Development is the story of one life. Evolution is the story of many lives across deep time. Another distinction that will matter later is the difference between anatomical change and less visible biological change. Anatomical change is what we can see directly in bones and bodies: the shape of the skull, the thickness of the brow ridges, the length of limbs, the size of jaws and teeth. This is the level at which anthropologists talk about anatomically modern humans, skeletons that fall within the range of shapes seen in humans living today. Less visible biological change can occur even when the skeleton looks almost the same. It includes differences in how our bodies process certain nutrients, how our immune systems recognize pathogens, how our blood responds to low oxygen at high altitude, or how we handle particular types of physical stress. Two people can look very similar on the outside and yet differ quite a lot in these hidden traits, many of which have a genetic basis. This distinction is important for our main question. When someone claims that humans stopped changing physically tens of thousands of years ago, they are usually thinking about big, visible anatomical changes, dramatic differences in skull shape, or overall body plan, like the differences between Homo sapiens and earlier human species. But a lack of dramatic anatomical change does not automatically mean that evolution has stopped. It may simply mean that much of the more recent evolution has taken place in physiology, metabolism, and the fine details of our biology, rather than in obvious new bones or body structures. If we go back to that small fire from the introduction, the camp about 50,000 years ago, it is easy to forget how long the road was that led there. Those people already look surprisingly familiar. But if we keep traveling backwards in time, generation after generation, their bodies and faces begin to change. At some point, we would no longer be looking at humans like us, but at earlier species that only hint at what we would eventually become. Human evolution starts with a split. Genetic and fossil evidence suggest that our lineage and the lineage leading to modern chimpanzees separated roughly 6 to 7 million years ago in Africa. The early members of our branch are often called hominins. Some of them, like Australopithecus afarensis, the species often associated with the famous skeleton Lucy, lived around 3.9 to 2.9 million years ago and already walked on two legs most of the time. even though their brains were still small and their bodies were quite different from ours. In other words, long before anyone we would recognize as human appeared, our ancestors were already experimenting with walking upright. Walking on two legs changed almost everything about the body. The pelvis had to become broader and more bowl-shaped to support the organs. The spine developed its characteristic curves to act as a spring. The foot gradually lost the grasping big toe seen in apes and became a strong platform for pushing off the ground. These changes took hundreds of thousands of years, but they created a new kind of creature, not yet human as we know it, but no longer a tree-dwelling ape either. A second major shift came with the rise of the genus Homo. Around 2 to 2.5 million years ago, fossils in Africa begin to show a combination of traits that we would associate with early humans: slightly larger brains, smaller teeth compared to earlier hominins, and stone tools in the same layers of sediment. Over time, this lineage produced Homo erectus, a species that appeared roughly 2 million years ago and survived in some regions until about 100,000 years ago. Homo erectus is often described as the first truly human-like body in our history. These individuals had longer legs, shorter arms, and body proportions closer to those of modern humans. They were capable walkers and probably capable of running over long distances. Fossils and stone tools show that Homo erectus did something no earlier hominin had done on such a scale. They left Africa and spread into parts of Asia and Europe. Their brains were still smaller on average than ours, and their skulls were more elongated with strong brow ridges. But the basic outline of a tall, mobile, ground-living human was already present. Between about 800,000 and 300,000 years ago, another stage unfolds, often called the Middle Pleistocene. In this period, we see fossils that are larger-brained than Homo erectus, but still more robust than us. Many researchers use names such as Homo heidelbergensis or archaic Homo sapiens for these populations. They are important because they seem to stand near the branching point between several later lineages: Neanderthals in Europe and Western Asia, Denisovans in parts of Asia, and the ancestors of modern humans in Africa. In Africa during this time, skulls gradually show a mixture of traits. Some are still quite robust, with heavy brow ridges, but others begin to display features that look more familiar. A face that is flatter and tucked under the brain case, smaller teeth, and a lighter-built skeleton. This mosaic is especially clear in a set of fossils from Jebel Haroud in Morocco, dated to around 300 to 315,000 years ago. These remains have a modern-looking, relatively flat face, but the back of the skull is more elongated, not yet the rounded, globular shape typical of many people today. Other important fossils come from Omo Kabish in Ethiopia and Florisbad in South Africa, dating to roughly 230 to 200,000 years ago. Together, these finds show that people with many recognizably modern traits were living in different parts of Africa well before the time of our imaginary campfire. Their faces and skeletons fall within the wide range we see in humans today, even though some details of the skull, especially the brain case, still look a bit archaic. This is where the term anatomically modern humans comes in. Anthropologists use it to describe fossils that fit within the range of body shapes and skull forms observed in living humans, as opposed to clearly archaic humans like Neanderthals. Anatomically modern humans are generally more lightly built, more gracile than earlier forms, with thinner bones, a higher and more vertical forehead, a reduced brow ridge, a face that is pulled under the brain case rather than projecting forward, and, in many cases, a distinct chin. It is important to see that this did not happen overnight. There was no single day or even a single 1,000-year slice of time when an archaic human woke up to find that their children were suddenly fully modern. Instead, different traits appeared at different times and in different places. Some fossils that are more than 300,000 years old already have quite modern faces, but archaic brain cases. Others, closer in time to 200,000 years ago, show a more modern skull shape, but still relatively robust bones. Human evolution in this period looks less like a straight line and more like a network, with different populations carrying different pieces of the modern pattern. By the time we reach the last 100,000 years in Africa, many of these pieces have come together. People who lived then would, in many cases, look familiar if we met them today. If you dressed them in modern clothing, most of them would not stand out on a city street. At the same time, other human lineages, Neanderthals in Europe and Western Asia, Denisovans in parts of Asia, and the ancestors of modern humans in Africa were evolving their own distinct versions of a large-brained, tool-using human. We now know from genetic data that when our anatomically modern ancestors finally expanded out of Africa, they did not simply replace these groups, they also interbred with them, leaving a small but measurable Neanderthal and Denisovan legacy in our DNA. By around 200 to 100,000 years ago in Africa, there were populations that we would comfortably call anatomically modern humans. Their faces, body proportions, and overall skeletons fell within the wide range we see in living people. But most of that story was still confined to one continent. The next step is not so much a new kind of body as a new kind of journey. Genetic and fossil evidence together suggest that modern humans left Africa more than once. There were earlier movements into the Near East over 100,000 years ago, and perhaps even small expansions beyond that, but most of those early groups seemed to have disappeared or contributed little to the DNA of people living today outside Africa. When we look at genomes from present-day Eurasians, Oceanians, and many other populations, a consistent pattern appears. Most of their ancestry traces back to a relatively small population that expanded out of Africa roughly 70,000 to 50,000 years ago. This is one reason why 50,000 years ago often appears as a kind of threshold in discussions of human evolution. It is not that nothing important happened before that date, but that a particular wave of humans, anatomically modern, behaviorally flexible, and carrying a specific set of genetic variants, began to spread widely and left a strong mark on the ancestry of people today. How exactly did they leave? Many researchers think that a major route out of Africa followed the southern coastline, crossing the narrow Bab el-Mandeb Strait at the southern end of the Red Sea, and then moving along the shores of the Arabian Peninsula, India, and Southeast Asia. Coastal environments could provide reliable food sources, such as shellfish and fish, and traveling along the shore may have been easier than crossing deep inland deserts and mountains. Over thousands of years, small groups moved eastward, sometimes splitting, sometimes rejoining, gradually spreading into new territories. One striking result of this movement is visible far from Africa. Recent genetic work on ancient and modern genomes from northern Australia suggests that humans were already present there around 60,000 years ago. This implies that not very long after leaving Africa, some groups had reached the shores of Southeast Asia and then crossed stretches of open water to enter the ancient continent of Sahul, which once connected present-day Australia and New Guinea. The people making these journeys were not sailors in the modern sense, but they already had enough technological and social skills to organize crossings on simple watercraft. At the same time, other branches of this expanding population were moving north and west. Fossils and genetic data indicate that modern humans were present in parts of Europe more than 45,000 years ago, overlapping with Neanderthals for several thousand years. In Western Asia and Central Eurasia, they encountered different climates and landscapes: steppe, forest, seasonal cold. Farther north, ice sheets advanced and retreated, opening and closing migration routes. Wherever they went, these travelers met not only new environments, but also other kinds of humans. Neanderthals had lived across Europe and Western Asia for hundreds of thousands of years. Denisovans, a more mysterious group, occupied large parts of Eastern Eurasia and possibly Southeast Asia. As our ancestors moved into these regions, they did not always keep to themselves. Multiple genetic studies now show clear traces of interbreeding between anatomically modern humans and at least these two archaic groups. For most people living today outside sub-Saharan Africa, about 1 to 2% of their genome comes from Neanderthals. The amount varies by region, and some populations in Oceania and parts of Southeast Asia carry additional contributions from Denisovans. In some groups, up to around 5% of their DNA can be traced back to them, with even higher levels in certain indigenous Philippine and Papuan populations. These are small numbers in percentage terms, but they are enough to show that when modern humans spread into Eurasia, they did not replace earlier humans overnight. For a time, different human lineages coexisted, interacted, and had children together. Interestingly, many of the genetic fragments we inherited from Neanderthals and Denisovans seem to be related to how our bodies cope with particular environments: immunity to local pathogens, response to sunlight, or adaptation to high altitude, among other things. In that sense, global dispersal and interbreeding did not radically reshape the human skeleton, but they did quietly modify the way our modern bodies functioned in new climates. If we look at the bones of early modern humans in Europe and Western Asia from about 45,000 to 30,000 years ago, they already fall within the range of anatomically modern humans. Some individuals are more robust, with slightly thicker bones or different details in skull shape, but none of them look like Neanderthals. They are tall, fully upright, with rounded skulls and relatively small faces compared to earlier humans. What changes, more than the basic form of the body, is the context around those bodies, the tools they make, the art they create, the ways they exploit local resources. In many archaeological sites from this period, we find finely worked stone blades, bone needles, ornaments made from shells and teeth, and a bit later, cave paintings and carved figures. These objects have often been used to argue for a creative explosion or a sudden rise of symbolic behavior in humans around this time, although newer discoveries in Africa and elsewhere suggest that the roots of these behaviors go back much further. Still, the combination of a widely spreading, anatomically modern body and a flexible, inventive mind is very visible in this phase of global dispersal. From a physical point of view, however, something interesting is happening. As these modern humans settle in very different parts of the world, in cold northern latitudes, in tropical forests, on dry grasslands, on coasts and high plateaus, the main outline of the body does not suddenly change into new, clearly separate species. Instead, we begin to see the early signs of regional variation within a single, globally distributed species. In colder climates, bodies tend to be slightly more compact, with relatively shorter limbs, which helps conserve heat. In warmer climates, longer limbs and a more linear build help to lose heat more efficiently. These patterns, later described as Bergmann's and Allen's rules, appear in many mammals, and humans are no exception. Over thousands of years, natural selection can gently shift average body proportions to fit local conditions, without changing the basic design of the species. So when people talk about humans stopping their physical evolution around 50,000 years ago, they are often looking at this big picture. By then, the major anatomical changes that distinguish us from earlier humans, our overall body plan, brain size, skull shape, and so on, were largely in place. The humans who left Africa in that main wave and spread across the world already looked very much like us. The differences that would gradually appear between populations in Europe, Africa, Asia, Oceania, and later the Americas were mostly variations on this same basic theme. From another angle, though, 50,000 years ago is not an endpoint, but a beginning. It marks the start of a new phase in which a single anatomically modern species occupies almost every kind of environment on Earth, mixes with other human lineages, and builds increasingly complex cultures. The bodies around that campfire at the start of our story carry within them both the deep history that led to modern humans and the potential for countless small changes still to come. By the time anatomically modern humans were spreading out of Africa and into the rest of the world, the basic outline of the human body was already familiar: a tall, fully upright figure with long legs, relatively small face, and a rounded skull. From a distance, someone from 50,000 years ago could probably walk past us on the street without attracting too much attention. This visual similarity is one reason people sometimes say that humans stopped changing physically at some point in the late Pleistocene. But this simple sentence can mean very different things, and not all of them are correct. One way to understand it is as a claim about anatomical shape at the species level. Compared with the strong differences between earlier human species, for example between Neanderthals and modern humans, the variation in skeletons of late Pleistocene and Holocene Homo sapiens is indeed relatively modest. Many studies of skulls and skeletons from the last 50,000 to 100,000 years show that they mostly fall within the range of what we see in living humans today, even if there are regional trends and historical changes in robustness or average size. In this limited sense, it is not completely wrong to say that the big anatomical changes had already happened by around the time our species began its main expansion out of Africa. A second way people use the phrase is as a claim about evolutionary forces. The argument goes roughly like this. Once humans invented complex culture, tools, shelters, clothing, agriculture, and finally medicine, we were no longer so strongly exposed to raw nature. Fewer people died from cold, starvation, or infection in childhood, so natural selection became much weaker. According to this view, we began to live outside of evolution, protected by technology. There is a grain of truth here as well. Many traditional causes of early death and infertility, such as severe infections, accidents during hunting, or deaths in childbirth, have been reduced in many societies. This changes the pattern of selection. Traits that once mattered a lot for survival might matter less, at least in some environments. But the strongest version of the claim that humans no longer evolve at all is not supported by the evidence. Genetic studies of modern and ancient DNA over the last 2 decades have shown clear signs of recent natural selection in our species. When researchers scan the genome for regions where certain genetic variants have rapidly increased in frequency, they find signatures of adaptation in the last tens of thousands of years and even in the last few millennia. Some of these signals are linked to physical traits, such as skin pigmentation, digestion of new foods, immune responses, or adaptation to particular climates. A well-known example is lactase persistence, the ability to digest lactose, the main sugar in milk, into adulthood. In most mammals, including early humans, the gene responsible for breaking down lactose is active in childhood and then largely switched off. In several human populations that developed dairy farming, genetic changes arose that kept this gene active throughout life. These changes spread rapidly over the last 5,000 to 10,000 years in parts of Europe, East Africa, and the Middle East, because adults who could drink milk had a nutritional advantage. Other clear examples come from high-altitude populations, such as Tibetans or Andeans, whose bodies showed genetic adaptations that help them cope with low oxygen levels, and from the immune system genes that bear the marks of strong selection in response to local pathogens. These changes are part of physical evolution, even if they do not create a new, easily recognizable body shape. So when someone says that humans stopped changing physically 50,000 years ago, they may be mixing together at least 3 different ideas: that no new human species have appeared since then, that the main outline of our anatomy has been relatively stable, and that evolution as a process has somehow ended. The first point is mostly true so far. We still have only one surviving species of human. The second point is partly true, in the sense that the differences between human populations in the last 50,000 years are smaller than the differences between modern humans and earlier species. But the third point, that evolution has stopped, does not fit the data at all. There is another subtlety here. From the fossil record alone, it can sometimes look as though nothing much is happening for long periods of time. A line of skeletons over tens of thousands of years can show what appears to be morphological stasis, little change in shape, especially if we are looking at relatively coarse measurements. In evolutionary biology, this pattern is known for many species and has led to ideas such as punctuated equilibrium, where long periods of relative stability in form can coexist with ongoing genetic and functional changes under the surface. Humans fit this picture quite well. The skeleton has not swung wildly into new designs in the last 50,000 years, but our genomes and some aspects of our biology have continued to shift. In some cases, even quite small genetic changes can have meaningful effects on how our bodies work in specific environments without producing a new type of human that would be obvious in a museum display. It also matters where we look for change. If we focus only on traits like overall brain size, limb proportion, or skull outline, we see a story of relative stability after the rise of anatomically modern humans. If we look at traits like disease resistance, metabolism, reproductive timing, or tolerance of certain diets, we see a more active landscape with different populations moving in different directions depending on their histories. In everyday language, all of this is easy to compress into the phrase, "we stopped changing physically." But in reality, what seems to have happened is more nuanced. The dramatic species-level anatomical shifts of earlier human evolution slowed down once a successful modern body plan had emerged, while finer-scale adaptations within that body plan continued, and in some cases may have even accelerated as our environments and lifestyles changed more rapidly. To untangle this further, it helps to step back and ask how, in earlier periods, the natural environment shaped human bodies in more obvious ways. Once we understand how climate, landscape, and food used to sculpt the human form, it becomes easier to see how the story changed when culture and technology began to shield us from some of those forces, and how, despite that, evolution carried on working in new directions. Long before cities, medicine, and global travel began to complicate our lives, human bodies were shaped very directly by the environments in which people tried to survive. Climate, sunlight, altitude, food, and disease all acted as filters. Those who happened to be slightly better suited to local conditions were more likely, on average, to leave descendants. Over many generations, these pressures left marks on our bodies, sometimes visible in skeletons, sometimes hidden in physiology and genes. One of the clearest examples is the relationship between climate and body size and shape. Biologists describe two patterns known as Bergmann's rule and Allen's rule. In simple terms, within a widespread species, individuals living in colder regions tend to have larger, more compact bodies, while those in warmer regions tend to have smaller, more linear bodies with relatively longer limbs. A larger, stockier body has less surface area relative to its volume, which helps conserve heat. A smaller body with longer limbs loses heat more easily, which is helpful in hot climates. Studies of fossil humans and of living populations show that temperature has been an important predictor of body mass and proportions over the last hundreds of thousands of years, in line with these rules, even though there are exceptions and local complications. If we compare traditional cold-adapted populations with groups from hot, open environments, this pattern becomes visible in everyday bodies, not just in statistics. People whose ancestors lived for many generations in cold, high-latitude regions often have relatively broad trunks and shorter limbs, which helps them retain heat. In contrast, populations whose ancestors lived in hot, low-latitude regions tend, on average, to have a taller, more slender build, with relatively longer arms and legs, which increases the surface area available for losing heat. These trends are not absolute rules. Individual variation and recent migration can blur the picture, but they illustrate how climate can nudge body proportions in different directions without turning one species into many. Another powerful environmental factor is sunlight, and especially ultraviolet radiation. One of the most visible ways humans differ is in skin color, and this variation is not random. Research by Nina Jablonski and others has shown that skin pigmentation strongly correlates with UV levels. Populations whose ancestors lived for a long time near the equator tend to have darker skin, while those from higher latitudes with weaker UV tend to have lighter skin. Dark skin, rich in melanin, acts as a natural sunscreen, protecting an important B vitamin called folate from being destroyed by intense UV radiation. At the same time, some UV is needed to produce vitamin D in the skin, which is essential for bone health and other functions. The evolution of human skin color can be seen as a balance between protecting folate and allowing enough UV through for vitamin D, with different solutions in different parts of the world. When our ancestors moved away from the intense equatorial sun into regions with weaker UV, especially at high latitudes with long, dark winters, heavily pigmented skin could become a disadvantage because it blocked too much UV for sufficient vitamin D production. In these environments, genetic variants that lightened the skin could be favored, and over many generations they spread. This is why light skin tones have evolved multiple times in different populations through different genetic pathways, rather than arising from a single origin. The pattern of human skin color around the globe is one of the clearest examples of the environment literally painting the human body. Altitude provides another example of how physical environments used to sculpt human biology. High-altitude regions, such as the Tibetan Plateau, the Andes, or parts of the Ethiopian highlands, present a serious challenge. The air contains less oxygen, which makes it harder to supply tissues and organs. Short-term visitors from low altitudes can adjust a little by breathing faster and producing more red blood cells, but long-term living at high altitude puts strong pressure on the body. Over thousands of years, populations native to these regions have evolved distinct physiological adaptations. Genetic changes in genes such as EPSA1 and EGLN1 in Tibetans, and different sets of changes in Andean and Ethiopian populations help their bodies cope with chronic low oxygen in ways that differ from the responses of recent arrivals. These adaptations are mostly invisible from the outside. A Tibetan or Andean person does not have a different species of body, but they show how a particular environment can shape the way a broadly similar human body actually functions. Food and disease are also parts of the environment, and they too have left marks on our biology. Pathogens have been among the strongest selective pressures in human history. Studies of immune system genes show many signatures of past natural selection, where variants that helped resist local infections became more common over time, even if they sometimes increased the risk of autoimmune diseases in different contexts. Likewise, changes in diet have shaped traits linked to digestion and metabolism. One widely discussed example is the number of copies of the AMY1 gene, which controls how much salivary amylase, an enzyme that digests starch, we produce. Populations with a long history of high-starch diets, such as some farming groups in Japan and Europe, tend to have more copies of AMY1 and higher levels of salivary amylase than populations whose traditional diets were low in starch. This suggests that, at least in some places, people whose bodies were slightly better at digesting starchy foods may have had a small advantage that accumulated across generations. Taken together, these examples, climate-related body shape, skin color tuned to sunlight, altitude adaptations, immune responses, and diet-related genes, show a world in which the physical environment constantly tested human bodies. In many cases, survival and successful reproduction depended on how well a person's inherited biology matched local conditions. For most of our history, there was little to soften these tests. If the climate was harsh, if food was scarce, if pathogens were deadly, there were not many tools to hide behind. What begins to change, gradually but profoundly, is not the fact that we have bodies in environments, but the nature of those environments. Fire, clothing, housing, tools, and later farming and medicine start to build a kind of cultural shell around the human body. Instead of simply adapting to the landscape, we begin to reshape the landscape around us. This does not mean that evolution stops. It means that many of the most important pressures on our bodies are no longer just cold, heat, or hunger, but also our own inventions. If it was cold, you needed a body that could conserve heat. If the sun was strong, you needed skin that could handle ultraviolet radiation. If the air was thin, your blood and lungs had to adapt. For most of our history, there was very little between the human body and the raw environment. With the growth of culture, that began to change. Humans did something unusual. Instead of only adapting to environments, we also started to reshape those environments around us, and then had to adapt to the worlds we ourselves had created. Biologists call this kind of process niche construction, when organisms, through their behavior, modify their own surroundings and, in doing so, change the selection pressures acting on them. In humans, culture makes this especially powerful. Our tools, buildings, social rules, and technologies create what some researchers call a cultural niche, a human-made environment that has become the main setting for our evolution. The earliest steps in this direction were simple but profound. Fire allowed our ancestors to stay warm in cold nights and to use new kinds of foods. Clothing made from animal skins and later woven fabrics added another layer of protection from weather. Shelters, even basic huts or caves with controlled fires, reduced direct exposure to wind, rain, and temperature extremes. All of this meant that survival depended a little less on having a perfectly climate-matched body and a little more on having access to the right knowledge and tools. The environment was no longer just the forest or the savanna. It included the camp, the fire, the shared skills of the group. At the same time, culture did not simply remove selection pressures, it also created new ones. As people became better at hunting and gathering, and later at storing food, some groups could support larger populations. Bigger groups meant more complex social structures, new forms of cooperation and conflict, and higher chances of spreading infections. Studies of human evolution over the past 40,000 years suggest that many strong episodes of selection are linked not only to natural environments, but also to cultural changes that altered diet, population density, and contact with pathogens. One of the clearest patterns where culture and genes interact is known as gene-culture coevolution. In simple terms, this means that a cultural practice creates new conditions for natural selection, which then favors genetic changes that make that practice even more successful, reinforcing both. A classic example is lactase persistence, the ability to digest the milk sugar lactose into adulthood. In most mammals, including early humans, the enzyme that digests lactose is switched off after weaning. But in populations that adopted dairying, keeping animals for milk, genetic variants that kept the enzyme active in adults provided a clear advantage, because they allowed people to use milk as an extra, relatively safe source of calories and nutrients. Over the last 5,000 to 10,000 years, these variants rose to high frequencies in parts of Europe, East Africa, and the Middle East. This is evolution driven by culture. Without the cultural practice of milking animals and drinking their milk, those genetic variants would not have been especially useful. With dairying, they became highly beneficial. The result is a feedback loop. Culture changes the environment. This environment favors certain genes. Those genes then help the culture to persist and spread. Similar feedback loops appear in other areas. When humans began to rely more heavily on starchy plant foods, for example, there is evidence that the number of copies of the AMY1 gene, which controls an enzyme that digests starch in saliva, increased in some high-starch populations, suggesting selection for more efficient digestion. When people started living in larger, denser settlements, especially after the rise of agriculture, they exposed themselves to new and more intense waves of infectious disease. Reviews of immune system genes show multiple signs that pathogens have been among the strongest selective forces in recent human evolution, with cultural changes such as crowding, animal domestication, and long-distance travel helping to shape which diseases were most important. Culture also alters how selection acts on reproduction. Social norms and economic systems can influence who has more children, at what age people form families, and how resources are passed on. Preferences for certain traits, sometimes physical, sometimes behavioral, or linked to status, can turn into a kind of cultural sexual selection, where the chances of having descendants depend not only on survival, but also on fitting into the expectations of a particular society. These effects are harder to measure than, say, altitude adaptation, but they remind us that, in a cultural species, the social environment is part of the evolutionary environment too. All of this does not mean that the natural environment stopped mattering. Even today, temperature, sunlight, diet, and disease still influence which traits are favored in different regions, especially where access to modern healthcare and technology is limited. But for at least the last tens of thousands of years, humans have been living in a layered world, the physical landscape underneath, and above it, a cultural layer of tools, buildings, foods, beliefs, and institutions. Our bodies are shaped by both layers at the same time. In some cases, culture softens old pressures. In other cases, it introduces new challenges that can be just as strong. To see this more clearly, it helps to focus on one of the most dramatic cultural shifts in our history: the transition from hunting and gathering to farming and settled life. This change did far more than alter how people got their food. It transformed diets, work, disease, social hierarchies, and the very rhythm of daily life. And with all of that, it added a new set of evolutionary tests for the human body. When we imagine progress in human history, agriculture and the rise of civilization often appear as clear steps forward. Farming brought permanent villages, surplus food, new tools, social complexity, and eventually cities and written language. It is tempting to picture early farmers as healthier and more secure than the hunters and gatherers who came before them. But if we look at their bones and teeth, the story is more complicated. The shift to agriculture, sometimes called the Neolithic Revolution, began in several parts of the world roughly 11,000 years ago, after the last ice age. In Western Asia, people domesticated wheat, barley, sheep, and goats. In China, rice and millet. In the Americas, maize, beans, and squash. Over thousands of years, many groups moved from a mobile life of hunting and gathering to more settled communities that depended heavily on a few cultivated plants and domesticated animals. From a demographic point of view, farming was a success. Fields and herds could support more people per square kilometer than wild plants and animals. Populations grew, villages expanded, and new forms of social organization appeared. But from the point of view of individual bodies, the first farmers often paid a price. Bioarchaeological studies comparing skeletons of foragers and early agriculturalists in many regions have repeatedly found signs of increased stress, disease, and in many cases, a decline in average stature after the adoption of agriculture. Height is a useful, if imperfect, indicator of childhood health and nutrition. It reflects the combined effects of genes, diet, and disease during growth. In many parts of the Old World, skeletons from early farming communities are shorter, on average, than those of earlier foragers from the same region, suggesting that children in the first generations of farmers often faced more nutritional stress or illness. More recent work with ancient DNA has added nuance, showing that in some areas the decline in stature was modest and influenced by shifts in ancestry as well as environment, and that later improvements in diet and living conditions could reverse the trend. Still, the general pattern is clear. For many early farmers, the new way of life was physically demanding and not always kind to growing bodies. Teeth tell a similar story. Foragers usually ate a varied diet of wild plants and animals, with relatively limited amounts of highly fermentable carbohydrates. Early farmers, in contrast, depended heavily on a few starchy crops, such as wheat, rice, or maize. As cereal-based diets spread, the frequency of caries, cavities, tends to increase in many archaeological samples. Caries form when bacteria in dental plaque feed on sugars and starches, producing acids that gradually dissolve tooth enamel. More sticky, starch-rich foods and more frequent eating create better conditions for these bacteria. In skeletal remains from early farming communities, archaeologists often find higher rates of cavities, abscesses, and tooth loss compared to earlier foragers, although there are regional exceptions. Bones also record the physical stresses of daily life. Evidence of stunted growth can appear as defects in tooth enamel or as growth arrest lines in long bones. Osteopenia and osteoporosis, signs of weaker bone tissue, as well as certain kinds of joint degeneration, become more common in some early agricultural populations, suggesting heavy workloads, repetitive tasks, and perhaps nutritional deficiencies, especially in childhood. Women in particular, in some early farming societies, seem to have carried a heavy double burden of physical labor and repeated pregnancies, with dental and skeletal markers of stress often more severe than in men. Disease patterns changed as well. Sedentary life in villages and towns meant more people living close together, often with poorer sanitation. The domestication of animals brought humans into continuous contact with new pathogens. Over time, several major infectious diseases, including forms of influenza, smallpox, and measles, are thought to have arisen or spread more easily in these crowded, farming-based societies. From the skeleton alone, it is not always possible to identify specific infections, but increased signs of chronic inflammation and certain types of lesions in bone are consistent with a heavier burden of disease in many early agricultural communities. All of this might sound like simple decline: more cavities, more infections, shorter bodies, harder work. Yet, evolution does not stop in such a setting. If anything, it gains new opportunities. The nutritional shifts, crowding, and exposure to animal-borne pathogens associated with agriculture created powerful new selection pressures on human immune systems and metabolism. Studies of ancient and modern DNA suggest that many genes involved in immune function, inflammation, and processing of different nutrients show strong signals of recent natural selection, especially over the last 10,000 years. In other words, agriculture did not simply make people sicker. It also began to sort human bodies in new ways. Individuals whose inherited biology could better handle high carbohydrate diets, frequent infections, or certain workloads would, on average, be more likely to survive and raise children. Over many generations, this kind of hidden sorting can reshape the frequencies of genetic variants related to digestion, immunity, and even growth, leaving a subtle but real mark on the bodies of later populations. So, when we look at the first farmers, we see a paradox. On the surface, their lives might look more advanced than those of foragers: fields, villages, pottery, stored grain. Inside their bodies, however, the early phases of agriculture often brought more stress, not less. But precisely because this new way of life was demanding, it created strong and partly novel pressures on the human organism. Our species had to adapt, not to a glacier or a savanna this time, but to dense settlements, limited diets, and close contact with herd animals. This is already a very different picture from the idea that human physical evolution stopped tens of thousands of years ago. The challenges simply changed shape. To see how clearly humans are still responding to those and later challenges, we can now zoom in on specific examples where recent evolution is visible almost in real time. In the genes that let some adults drink milk, in the bodies of high-altitude populations, and in the continuing reshaping of our immune defenses. Up to this point in the story, I have mostly been talking about long time scales, millions of years from early hominins to modern humans, tens of thousands of years from the first fires in Africa to the spread of our species across the world. It is easy to imagine that evolution belongs only to that deep past. But when we look closely at our genes and at certain traits in modern populations, a different picture appears. Some of the clearest signals of natural selection in humans are not ancient at all. They belong to the last 10,000 years, and in some cases may even still be active today. One of the most famous examples is the ability of some adults to digest the sugar in milk. Like all mammals, human babies are born able to break down lactose, the main sugar in milk. In most mammals, this ability is switched off after weaning. For our distant ancestors, this was the default pattern. Children could drink milk, adults could not. But in several human populations, a genetic change occurred that kept the lactase enzyme active into adulthood, a trait known as lactase persistence. On its own, this change would not have been particularly important. What made it powerful was a cultural invention, dairying. When people in parts of Europe, the Middle East, and Africa began to keep cows, sheep, or goats for milk, they created a new, rich source of food. For adults who carried lactase persistence variants, fresh milk could be safely digested. For those without them, too much milk could cause digestive problems. Genetic studies show that in some European and East African groups, the DNA variants associated with lactase persistence rose from rarity to high frequency in just the last 5 to 10,000 years, a speed that implies very strong natural selection. This is not a subtle shift at the level of a small metabolic detail. It is a clear change in what adult bodies can do with a common food, driven by a specific cultural practice and leaving a strong signature in our genomes. If humans had truly stopped evolving physically tens of thousands of years ago, we would not see such a rapid, recent transformation in something as basic as digestion. A second, equally striking example comes from life at high altitude. Places such as the Tibetan Plateau, the high Andes, or the Ethiopian highlands present a challenge that is directly physical. The air is thinner, and there is less oxygen. Short-term visitors from low altitudes respond by breathing faster and producing more red blood cells. But over the long term, this response can itself become harmful, leading to thick blood and health problems. Populations that have lived at high altitudes for thousands of years show a different pattern. Tibetan highlanders, for instance, typically have only moderately elevated hemoglobin levels compared with lowlanders, rather than the very high levels seen in recent arrivals. Genetic studies have identified variants in genes such as EPAS1 and EGLIN1, which are part of the body's response pathway to low oxygen that are common in Tibetans and show strong signs of recent positive selection. These variants appear to fine-tune how the body reacts to chronic hypoxia, reducing some of the harmful effects of living at altitude. Andean and Ethiopian high-altitude populations have evolved their own, partly different sets of adaptations, using other genetic routes to solve the same environmental problem. Here again, we see evolution operating within an already modern human body plan. The skeleton of a Tibetan villager does not belong to a different type of human than that of a lowland farmer, but the way their blood and lungs respond to the same air can differ in important inherited ways. These changes are recent on an evolutionary timescale and tied directly to a physical challenge in the environment. A third line of evidence comes from the long battle between humans and infectious diseases. Malaria, in particular, has left deep marks in the genomes of populations living in regions where the disease is common. One classic case is the sickle cell trait. A single change in the gene for hemoglobin produces a version of the protein that, when inherited from both parents, leads to sickle cell disease, a serious and often life-shortening condition. But when inherited from only one parent, in other words, when a person has one sickle allele and one normal allele, it provides significant protection against severe forms of malaria. Because of this, in regions where malaria has been intense for many generations, the sickle allele has been maintained at relatively high frequencies through a form of balancing selection. It is harmful when doubled, but advantageous when present in a single copy. The geographical overlap between areas of historically high malaria transmission and the distribution of the sickle cell allele is striking, and there is good evidence that malaria has continued to favor this allele in parts of Africa even in recent centuries. Sickle cell is not the only example. Variants in other genes related to red blood cells and immune function also show signs of selection in malaria-endemic regions. And different diseases have shaped other parts of our genetic landscape. One widely discussed case is the CCR5 delta 32 mutation, a deletion in a gene that encodes a receptor on immune cells. Individuals who carry two copies of this deletion are highly resistant to infection by HIV, and those with one copy may progress slowly to AIDS. Genetic and modeling studies suggest that the allele rose to relatively high frequencies in parts of Europe within the last 1,000 years under selection from past pathogens, possibly smallpox, although the exact history is still debated. Together, these medical and genetic stories reveal a simple truth. Our bodies continue to be shaped by the challenges around us. Sometimes the challenge is a new food source, like milk. Sometimes it is the thin air of high mountains. Sometimes it is a parasite carried by mosquitoes or a virus spread through crowded communities. In each case, small genetic differences between individuals can make a real difference to survival and reproduction. Over many generations, those differences can change the average biology of a population. It is worth noting that not every recent change in human bodies is evolutionary. Some trends, such as increases in average height or earlier ages at puberty in many countries over the last century, are largely explained by improved nutrition, healthcare, and living conditions, not by changes in genes. But the presence of strong detectable signals of natural selection in our genomes tied to concrete physical traits shows that evolution has not switched off. It has simply become more entangled with our cultural choices, our technologies, and our diseases. Seen from this angle, the idea that humans stopped changing physically 50,000 years ago becomes very hard to defend. The big outline of our skeleton settled into its modern form by that time, but within that outline, evolution has continued to adjust how our bodies digest food, carry oxygen, respond to infections, and perhaps handle other aspects of life in a changing world. The interesting question today is not whether evolution is still acting on us, but how its direction and intensity have been altered by the very tools we use to protect ourselves, especially medicine and public health. When people say that humans have stepped outside of evolution, they usually have one simple idea in mind. Modern medicine and technology save lives that would once have been lost. A baby born prematurely, a child with a serious infection, an adult with a heart condition. Many of these people now survive and have children of their own thanks to vaccines, antibiotics, surgery, and intensive care. In earlier centuries, they might not have reached adulthood at all. It feels natural to think that if almost everyone survives, natural selection must have faded away. To see what has really changed, it helps to remember how strong selection on survival used to be. In many pre-industrial societies, a large fraction of children died before the age of 5, often from infections, malnutrition, or complications around birth. Historical demographic studies show that under 5 mortality rates in many countries were extremely high until the 19th and early 20th centuries, and have since fallen dramatically, often by a few percent per year over long periods. For most of our history, simply living long enough to reproduce was a major hurdle, and any inherited trait that slightly improved survival in that harsh environment could be strongly favored. Over the last century and a half, this picture has changed. Clean water, sewage systems, vaccines, antibiotics, safer childbirth, and better nutrition have sharply reduced deaths in childhood in much of the world. In many countries today, the majority of children survive to adulthood, and many causes of early mortality that once shaped our genomes have been weakened. Researchers who study relaxed selection argue that when fewer people die before reproducing, natural selection removes fewer harmful genetic variants each generation, allowing more mildly deleterious mutations to persist in the population. On this view, modern healthcare has softened some of the harshest filters that once acted on the human body. There is some support for this idea at a genetic level. Comparative studies of mutation load, the accumulation of slightly harmful genetic changes, suggest that relaxed selection in recent human history may have allowed more of these variants to remain in our genomes, especially in populations with very low child mortality and high-quality medical care. However, these changes are subtle and unfold over many generations. At the same time, almost every measurable indicator of health and survival has improved. Life expectancy has more than doubled over the last 150 years, child mortality has collapsed in many regions, and average height has risen where nutrition improved. In other words, if relaxed selection is slowly increasing the number of slightly harmful mutations, its effects are currently outweighed by better environments, medicine, and nutrition. The other important shift is that selection has moved its weight from survival to reproduction. Natural selection does not stop just because most people live to adulthood. It operates whenever individuals differ in how many children they have, and when those differences are linked, even weakly, to inherited traits. As evolutionary biologists like Stephen Stearns have pointed out, the potential for natural selection only truly vanishes if everyone has exactly the same number of children, and no heritable trait is correlated with reproductive success, a situation that is extremely unlikely in any real human population. Modern societies show large differences in fertility between individuals and between groups. In many high-income countries, total fertility rates have fallen below replacement level, and people with higher education or income often have fewer children and start families later than those with lower education or income. Recent genetic studies using large biobanks have found that polygenic scores associated with higher education, higher earnings, or certain health traits tend, on average, to be associated with slightly lower numbers of children, whereas some other traits show the opposite pattern. The effects are small from one generation to the next, but they are real examples of ongoing natural selection in contemporary populations, operating through differences in fertility rather than through dramatic differences in survival. At the same time, medicine itself can create new selection pressures. Treatments that allow people with particular conditions to survive and reproduce mean that the genes underlying those conditions can persist and spread. From a narrow genetic perspective, this can increase the prevalence of certain risk variants. But from a human perspective, this is exactly what medicine is for: to reduce suffering and give people a chance at a full life, regardless of their genetic luck. Some researchers have even suggested that in future, gene therapies and reproductive technologies could partly replace natural selection by directly editing or choosing certain traits, although these ideas are still more speculative than real. So, medicine and technology do two things at once. They relax some of the old forms of selection, especially brutal childhood mortality from infection or malnutrition. They also open space for new patterns of selection, mainly through differences in who has children, at what age, and under what social conditions. On top of that, they create a new kind of cultural filter. Access to healthcare, contraception, education, and assisted reproduction all shape which bodies and which genes are more common in the next generation. From the viewpoint of our main question, this means that evolution has not stopped. The rules have shifted. Where survival once depended heavily on matching the climate, finding food, and resisting infections with no help, it now depends more on access to medical systems and the social and economic structures around us. Where selection once fell hardest on whether a child lived to age 5, it now acts more quietly through the timing and number of births and through how different traits are linked, genetically and socially, to the choice or ability to have children. If we put this together with the examples from the previous chapter, lactase persistence, altitude adaptations, resistance to diseases like malaria, a pattern emerges. Humans have moved from a world where nature was the main sculptor of bodies to a world where nature and culture carve together. In that world, not only medicine, but also patterns of movement, marriage, and migration matter. If we jump from our imaginary campfire 50,000 years ago to a view of the world today, one of the most striking differences is how thoroughly human populations are now connected. People, ideas, and genes move across continents in ways that would have been impossible for almost all of our history. Flights cross oceans in hours. Millions of people live far from the places where their grandparents were born. Cities bring together neighbors with very different family histories. It is natural to wonder what all of this movement is doing to human physical diversity. Are we slowly blending into one global population, Or is the picture more complicated? Genetic studies over the last few decades have revealed two important facts that form the background to this question. The first is that humans are a remarkably homogeneous species compared with many others. When population geneticists measure how genetic variation is distributed within and between groups using statistics such as FST, they consistently find that most variation, often around 85 to 90%, exists within local populations, with only a small fraction, roughly 10 to 15%, between major geographic groups. This means that two people from the same village can be genetically quite different from each other, and two people from different continents can sometimes be relatively similar, depending on which variants we look at. The second fact is that, despite this overall similarity, human genetic variation is not randomly spread across the globe. There are patterns or structure reflecting past separations and later connections between populations. Studies that analyze hundreds of genetic markers in individuals from many regions find that people tend to cluster by geography, with neighboring groups more similar on average than distant ones. These patterns are blurred at the edges, but still visible, like colors bleeding into each other on a map rather than sharply separated blocks. What globalization has changed is the speed and extent of mixing between previously more isolated groups. Population geneticists use the term admixture for this process. Gene flow between populations that were once more distinct. Recent reviews point out that admixture is not a rare exception, but a common feature of human history. Most modern populations show evidence of ancestry from multiple sources, reflecting waves of migration, conquest, trade, and enslavement. In places like the Americas, for example, genome-wide studies reveal complex combinations of indigenous American, European, and African ancestries, shaped by colonization, the Atlantic slave trade, and later migrations, with unequal contributions and sex-biased patterns that are still being disentangled today. What is new in the last few centuries is not admixture itself, but its scale and the cultural context. Massive urbanization and labor migration have created cities where neighbors may have roots on different continents. According to recent United Nations and World Bank estimates, more than half of the world's population now lives in urban areas, and that fraction is projected to rise further with especially rapid city growth in parts of Africa and Asia. In these environments, everyday social life, schooling, and work bring together individuals whose ancestors might, a few hundred years ago, have had almost no contact. From a genetic point of view, sustained mixing over many generations tends to reduce differences between populations. If people from distant regions have children together, the resulting offspring carry combinations of genetic variants that previously rarely met in the same body. Over time, this gene flow can lower FST values between groups and smooth out sharp boundaries in genetic structure, especially where barriers to intermarriage weaken. In that sense, continued globalization does push human populations toward greater genetic connectivity. However, This does not mean that all human bodies are on their way to becoming identical. First, mixing is uneven. Some regions and social groups are highly connected, with frequent migration and intermarriage. Others remain relatively isolated by geography, language, religion, or politics. Second, new forms of structure can emerge even as old ones fade. If people tend to choose partners from similar social, cultural, or economic backgrounds, assortative mating can maintain or create patterns of genetic correlation that do not map simply onto geography. Third, even in a highly connected world, local environments, both natural and cultural, can still favor certain traits over others. Climate, altitude, diet, and disease burdens are not identical everywhere, and they may continue to shape subtle physical differences between populations through natural selection, as we saw in earlier chapters. At the same time, cultural practices, economic systems, and health policies differ between countries and communities, influencing who has more children and under what conditions. These forces can maintain or even create new gradients in traits, even against a background of general mixing. One area where globalization's impact is especially visible is the distribution of traits related to disease risk and drug response. Medical genetic studies increasingly show that risk variance for complex diseases and the frequencies of alleles that influence responses to medications vary across populations, shaped by both ancestry and recent history. As admixture proceeds, these distributions are being reshuffled, sometimes reducing the prevalence of certain risk alleles in particular groups, sometimes introducing them into new contexts. For clinicians and public health planners, this means that simplistic assumptions based on broad racial categories are less and less useful. Attention has to shift toward individual genetic profiles and more precise measures of ancestry. From the perspective of our story, the key point is that globalization is not ending human diversity, it is reorganizing it. The old picture of neatly separated races with deep, fixed biological boundaries never really fit the data, but it becomes even less accurate in a world where people with different ancestries meet, live, and have children together on a large scale. At the same time, the idea that all humans will soon look and function the same also misses something important. Even in a more mixed world, variation in genes, bodies, and environments will remain, and evolution will continue to work with that variation. In everyday conversation, it is easy to slide into extremes. Either we are all the same now, or we are divided into fundamentally different kinds of humans. Modern genetics suggests a quieter middle ground. Most human variation has always been found within populations. Differences between regions are real but modest, and recent mixing is knitting the species together even more tightly, without erasing the fine-grained patterns that make every population and every person slightly unique. By this point in the story, the main message is already clear. Humans did not step out of evolution 50,000 years ago. Yet when people talk about our species today, a few simple stories still come up again and again. They are attractive because they make a complicated process sound easy, but they do not fit what we know from fossils, genetics, and modern populations. One very common claim is that human evolution has stopped because medicine and technology protect almost everyone, so natural selection has nothing left to do. As we saw in the previous chapter, there is a kernel of truth hidden here. In many countries, child mortality has fallen dramatically in the last century. and some of the most brutal filters on survival have been weakened by clean water, vaccines, and healthcare. But the stronger version of the claim that evolution is over is not supported. Evolution does not require high death rates. It requires heritable variation and differences in reproductive success. As long as people differ, even slightly, in how many children they have, and some of those differences are linked to inherited traits, natural selection can still act. Reviews of contemporary human evolution make exactly this point. Medicine may relax selection on survival, but it does not remove selection altogether. Genomic data reinforce this. When researchers scan human genomes, both modern and ancient, they find many regions where certain genetic variants have spread unusually fast in the last 10,000 years or so, a classic sign of recent positive selection. Some of these regions are associated with traits we have already met in this story: the ability to digest milk in adulthood in dairying populations, genetic changes linked to high-altitude life on the Tibetan Plateau or in the Andes, and variants in immune system genes shaped by malaria and other infectious diseases. These signals show that evolution has continued to modify human bodies well after anatomically modern humans had spread across the globe. If evolution were truly over, such patterns would be hard to explain. A second misconception pulls in the opposite direction. If evolution is still happening, some people imagine that it must be driving us toward being more advanced or better humans, perhaps taller, more intelligent, stronger, or longer-lived. This idea quietly assumes that evolution has a built-in direction or goal, like a staircase with primitive at the bottom and advanced at the top. In modern evolutionary biology, that picture does not hold. Natural selection does not aim at an abstract improvement. It simply favors traits that increase reproductive success in a particular environment, even if those traits would be neutral or harmful in another setting. The history of farming is a good reminder of this. Agriculture allowed larger populations and more complex societies, but early farmers were often shorter, sicker, and more stressed than nearby foragers. From an evolutionary point of view, this was not a mistake, it was a trade-off. A lifestyle that increased the number of surviving children could spread even if it made individual lives harder. The same is true today. Traits that help in one social or economic environment might be a disadvantage in another. Evolution is about local fit, not about a universal ranking of better and worse humans. A third myth concerns human differences between groups. People often talk as if our species were divided into a small number of deep, natural, biological kinds, races, that differ from each other almost like subspecies in other animals. From this perspective, it becomes easy to slip into talk of certain races being more or less evolved. Genetic studies over the last 50 years tell a different story. Starting with Richard Lewontin's work in 1972 and later confirmed by many others, researchers have repeatedly found that most human genetic variation lies within local populations, not between large geographic groups. Roughly 80 to 90% of genetic differences are between individuals in the same broad group, with only a modest fraction between continents. This does not mean there is no structure at all. When thousands of genetic markers are analyzed together, individuals tend to cluster in ways that reflect geography and history, and neighboring populations are usually more similar than distant ones. But the boundaries between these clusters are fuzzy, and variation changes gradually across space rather than jumping in sharp steps. Most geneticists and anthropologists therefore argue that traditional race categories are poor descriptions of human biological diversity. They mix together many different patterns into a few broad labels and invite misleading stories about some groups being more evolved than others, when in reality all living humans share a recent common origin and a high degree of overlap in their genetic variation. Another misconception grows out of our increasingly connected world. As people from different regions meet and have children together, some observers imagine that this mixing will quickly erase human diversity and bring evolution to a halt. It is true that gene flow between populations tends to reduce sharp differences between them, and current admixture is knitting previously separated groups together even more tightly. But mixing does not remove variation, it reshuffles it. Children of parents from different backgrounds carry combinations of genetic variants that previously rarely met in the same body. Natural selection, if it acts on any of those combinations in particular environments, can still change their frequencies over time. Rather than ending evolution, a more mixed world simply means that future evolutionary change will start from a different, more interconnected pattern of variation. A final and very influential myth is that humans have switched from biological evolution to purely cultural evolution, that culture has replaced genes. It is certainly true that culture evolves very quickly. Ideas, technologies, and social norms can change within a single generation, far faster than genetic evolution. But culture does not float above biology. It is part of the environment in which our genes operate. As several researchers have argued, culture has become one of the main drivers of recent human evolution, through processes of gene-culture co-evolution. The ability of some adults to digest milk is only useful in cultures that keep dairy animals. High-altitude adaptations matter only for groups that live at altitude. Changes in immune genes are shaped by patterns of settlement, animal domestication, and travel. Our cultural choices create new selective pressures, which in turn can favor genetic changes that make those choices easier or more advantageous. Taken together, these myths have something in common. They either freeze evolution in the distant past, as if nothing important has happened since humans began to look like us, or they turn evolution into a story about ranking human groups, with some imagined as ahead and others behind. The picture that emerges from fossils, genetics, and anthropology is quieter but more interesting. A single, recently evolved species, with most of its variation shared and shuffled within and between populations, still responding to changing environments, many of them of our own making. With these misunderstandings cleared away, it becomes easier to ask a more speculative but natural question. If our bodies have not stopped changing, what kinds of physical changes might lie ahead as climate, technology, and social life continue to shift? Anytime we ask about the future of human evolution, we are stepping into guesswork. We cannot know which technologies will truly take hold, which societies will flourish or collapse, or what surprises nature still has for us. What we can do is take the same forces we have seen in the past, natural selection, gene culture co-evolution, migration, and drift, and ask how they might work in the kinds of environments we are building now. The answers will not be precise predictions, but they can show us the kinds of changes that are plausible. One obvious driver is the physical environment, especially climate. Climate change is already affecting human health and survival through more intense heat waves, changing patterns of infectious disease, food and water stress, and extreme weather events. Assessments by the IPCC conclude with high confidence that climate change has already harmed physical and mental health, and that heat-related illness and mortality are rising as temperature extremes become more common. In the short term, societies can respond with technology and policy, air conditioning, better housing, heat wave warning systems. But if certain regions become consistently hotter and more humid, there may also be evolutionary pressure on traits linked to heat tolerance, sweat and salt balance, kidney and cardiovascular function. These shifts would probably be subtle and difficult to separate from cultural responses, but over thousands of years, they could leave a trace in the bodies of people living in the hottest, most stressed environments. A second major trend is urbanization. For most of our history, humans lived in small, scattered groups. Today, more than half of the world's population lives in urban areas, and United Nations projections suggest that this share may grow to around 2/3 by 2050. Cities concentrate noise, pollution, social stress, artificial light at night, and limited access to green spaces, but they also concentrate healthcare, education, and economic opportunities. Some researchers describe humans as an increasingly urban species, still learning how to cope biologically with the dense, fast, noisy environments we have created. Urban life brings a different pattern of risks than the one our bodies evolved in. In many countries, infectious diseases and undernutrition are no longer the main threats. Instead, chronic conditions like cardiovascular disease, type 2 diabetes, and some cancers are linked to diets rich in energy-dense foods, low physical activity, and long hours spent sitting. Global analyses report that obesity rates have roughly tripled since the 1970s, with over 1 billion people classified as obese in 2022, and further increases projected. These changes are happening far faster than genetic evolution, so most of the current patterns are driven by behavior and environment rather than by new mutations. But over tens of thousands of years, if certain lifestyles and disease risks are stable, and if they consistently affect who has more surviving children, they could shift the frequencies of genetic variants related to metabolism, appetite, or cardiovascular resilience. At the same time, global mobility and mixing are reshaping the raw material on which evolution can act. As we saw earlier, large-scale migration and intermarriage are combining genetic variants from populations that were once more separated. In a distant future, if mixing continues, we might see fewer sharp regional differences in traits like skin pigmentation or average body shape, and more fine-grained variation within a broadly interconnected global population. Natural selection would then act on this blended background, perhaps favoring different combinations of traits in different local environments, but with less of the deep geographic separation that characterized much of our past. Technology adds another layer of uncertainty. For now, most medical advances change selection indirectly by reducing deaths and altering who can have children. But genetic technologies, from embryo screening to potential germline gene editing, raise the possibility that future humans might not only be shaped by natural selection, but also by deliberate choices. CRISPR-based genome editing has already been used experimentally in human embryos, and there is an active ethical debate about whether and when it might be acceptable to make heritable changes aimed at preventing disease or altering other traits. Strict regulations and social norms currently limit such interventions, and it is not clear how widely they will ever be used. Still, if, over many generations, some societies apply genetic technologies systematically while others do not, it could create new patterns of biological difference that are partly planned rather than purely evolved. Looking out as far as 50,000 years, it is tempting to imagine dramatic transformations. Humans with vastly enlarged brains, or physically adapted to life in space, or split into different engineered types. Science fiction explores these possibilities freely. Real evolutionary change, however, tends to be slower and more conservative, especially in a species as widespread and mobile as ours. Unless humanity fragments into long isolated groups with very different environments and little contact, it is unlikely that new human species will appear on this timescale. Our descendants may still be recognizably Homo sapiens in their bones, just as we recognize people from tens of thousands of years ago as one of us. The more realistic picture is one of continuity with variation. Future humans will still inherit genes from their parents, develop in wombs, grow up in bodies that must balance energy, temperature, disease, and reproduction. But the pressures acting on those bodies will not be the same as those that shaped our ancestors on the savanna or in early farming villages. Climate will be warmer and less predictable. Most people will live in cities. Diets, sleep patterns, and social networks will be governed by technologies we are only beginning to see. Under those conditions, evolution will continue to adjust the fine details of our biology, perhaps shifting disease risks, stress responses, or metabolic settings, even if our overall appearance changes only modestly. In a way, we have come full circle. 50,000 years ago, humans whose bodies were already modern in shape stepped into new worlds as they left Africa and met unfamiliar climates and other human lineages. Today, humans whose bodies are still recognizably the same are stepping into new worlds of climate change, dense cities, and powerful technologies. In both cases, the species is moving faster than evolution can, but evolution follows along, working with whatever variation is available. If you picture again that moment at the beginning of our story, you waking up 50,000 years ago by a small fire under the night sky, the people around you no longer feel like mysterious strangers. By now you know, in some detail, what had to happen for them to exist. Millions of years of slow change from tree-dwelling apes to upright walkers, from small-brained hominins to large-brained humans, from robust, archaic bodies to anatomically modern ones. You also know that, At the level of bones and basic proportions, they are already very close to us. If one of them stood up, walked into the 21st century, and put on modern clothes, we would probably recognize them as just a person. Our skeletons, our general body plans, even the way we move and gesture, are part of the same design. That is why it is so tempting to imagine that this design was somehow finished long ago, fixed in place by evolution, and then simply copied forward into the present. But as we walked through the chapters of this story, a different picture emerged. What seemed at first like an endpoint, the appearance and global spread of anatomically modern humans, turned out to be more like a bend in the road. Once the overall shape of the human body had stabilized, evolution did not stop. It shifted its focus. Instead of building a completely new frame, it began to work more quietly inside that frame, tuning digestion to new diets, adjusting blood and lungs to thin air, reshaping immune responses to changing disease landscapes. At the same time, something else was happening. Humans were no longer just bodies in nature, we had become bodies in cultures. Fire, clothing, and shelters softened some of the old pressures from climate. Farming concentrated people and their animals, creating new pressures from diet and disease. Cities and global travel rewrote the patterns of who meets whom, who shares genes with whom, and which traits help or hinder raising children. Evolution did not vanish in this cultural world, it gained a new partner. Genes and culture began to shape each other, sometimes gently, sometimes very strongly. This is why the idea that we stopped changing physically around 50,000 years ago does not quite fit. It is true that no new human species have appeared since then, and that the differences between human populations today are small compared with the gap between us and Neanderthals or earlier hominins. It is also true that modern medicine and technology have relaxed some of the harshest forms of natural selection, especially deaths in early childhood from infection or hunger. But within those broad outlines, the evidence for continued change is everywhere, in the genomes that carry traces of recent selection, in the bodies of high-altitude peoples and dairy farmers, in the complex mix of ancestries that now meet in global cities. If we look ahead, the forces acting on human bodies are not slowing down. They are simply different from those our ancestors faced. A warming climate, dense urban life, new diets, new disease risks, and powerful technologies all create environments that our species has never experienced before. Some of the responses will be cultural and technological, unfolding quickly. Changes in behavior, policy, medicine. Others will be slower and biological, happening across generations, often too gradually to notice in a single lifetime. Both belong to the same story. So what does it mean to say that humans never stopped changing? It means that evolution is not something that happened to other kinds of humans long ago, leaving us as finished products. It is an ongoing process, working with small differences between individuals in every generation, influenced now as much by our own inventions as by climate or geography. Our bodies carry the marks of ancient savannas and ice ages, of fireside stories and plowed fields, of epidemics and migrations, and quiet everyday choices about food, family, and work. If you return, in imagination, to that person by the fire 50,000 years ago and sit down beside them, you are sharing much more than a romantic moment in deep time. You are sharing a continuous line of change that runs through them, through you, and on into people who have not yet been born. From their point of view, we are the strange ones, taller or shorter, living longer or faster, surrounded by machines, carrying in our blood and bones the consequences of histories they could not imagine. And just as we can look back and see how their world still lives in us, someone in the far future could look back at us and see the early traces of changes that for them will feel ancient and obvious. In that sense, we are not the end of the human story. We are just another turning point, another group of people sitting around our own kinds of fires, sometimes real, sometimes the light of a screen, while the quiet work of evolution continues in the background, shaping bodies that are always recognizably human and never exactly the same. Thank you so much for watching our video and supporting the channel. See you in the next episodes, and good night.